Can you all hear me well? Does that work? Okay, lovely. Nice. So we'll have roughly 3 hours, and in those 3 hours, we're going to talk about wet lab, AI, and data, and the interplay between these three. And this is particularly based on our experience on working together on a protein engineering project. Me personally from the AI side of things, and Max on the wet lab side of things. And I think the relevance of AI in the biological sciences and the sciences at large was underscored very impressively last week when the Nobel Prize for AlphaFold and protein design went to these three individuals who all three kind of contributed to the use of AI in biology at large. And so to me, that's kind of a sign that AI is coming of age in biology. And so we were, over the course of our PhDs, we've always been kind of wondering what's the best way of working together as someone who comes more from the computational side of things and someone who comes more from the wet lab side of things. And so given this as a primer of relevance, this is the rough menu for the afternoon, what we've planned for you today. There's going to be a very brief intro. Who are we and who are you? And then we'll do a whirlwind tour through AI and data, a couple of definitions, a couple of just insights, rough ideas and thoughts around data. We'll take a brief coffee break, and then we'll go into a deep dive on a real-world case study. So the wet lab AI collaboration between Max and myself and the team we've been working with. And we'll specifically also talk about the human side of AI wet lab collaborations and what it meant for us working together. And then we'll have Matthew Penna, who'll be with us in 20 minutes, joining to also talk about his experiences briefly, and then go for another coffee break. And finally, I think the most exciting part, to interactively get into your projects and your research and see what might be applicable of the things we've been talking about, and also how we can all benefit from each other's knowledge in the room, because I think many of you have very likely also had quite a few experiences and can learn from each other. So that's the agenda. And with that, without further ado, who are we, your AI traffic controllers for today? Max, I'll let you take it from here.
Yeah, exactly. I hope you all hear me well as well. Yeah, that works, perfect. So my name is Max. I am currently still a PhD student at the Department of Biochemistry in Cambridge. My training is completely from the wet lab side of things. Concerning a lot of things that you'll hear today, I'm very new to the kind of hard coding of AI models. And my role here is a little bit to be the scientists that Simon collaborated with, and that can point you to a few things we've learned along that journey. When it comes to data, the limitations of data, and how you interact with your AI collaborator to make them aware of your limitations and to be aware of their limitations. We'll have a guest speaker around, Matt Penna, who is also supervised by Florian and who has had similar experiences in the protein design kind of world. Maybe Simon, you want to say something about yourself quickly?
Yeah, I'm a PhD student in the Cambridge Computer Lab. I have not got any wet lab experience. Max has kindly explained to me, how things work, and it's been a very exciting journey and very relevant also to the computational work, because many decisions on the modeling side of things and how to treat various pieces of data that came back from assays that Max ran really crucially required your input in order to actually make progress meaningfully.
Yeah, and diving deep into kind of what we are interested in and what kind of our research area is, we both are interested in sustainability, and that really drives both our research. And what drove me into doing a PhD in that area was looking at this enormous complexity that we have when it comes to small molecules and in the natural world, right? If you look at those incredible small molecules, chemists out there will say impossible, we cannot make those. We cannot synthesize them in the lab. And in fact, actually, for this drug here, Taxol, which is a natural product, one route to making this drug involves 25 steps in synthesis. You start with hundreds of kilograms of substrates, and you end up with a tiny little amount of that product. So you actually have to source this from the environment. Nature, however, makes those small molecules completely naturally using life's chemists, using enzymes, proteins that catalyze those reactions that would be basically impossible to do using classic organic chemistry. And nature does this in a very natural, sustainable way, right? If you look at plants, they only use sustainable feedstocks. Everything works in water. At moderate temperatures, basically, as the chemists would say it, in one pot, right? We put everything together inside one cell. We can put everything into a cascade, and then this cell synthesizes the small molecule that you want. And not only do we start with renewable starting materials, so we don't need any petrochemicals, but we can make every transformation that we like stereoselective. What does stereoselective mean? We can, make a bond point exactly in the right direction as we want this bond, and chemical synthesis, organic synthesis is not very good at this. And finally, those enzymes are also evolvable. That means we can change the genetic code, incorporate an amino acid mutation at a position that we'd like, and just change the way those enzymes work. So quite naturally, enzymes are very, very good at their job. And this has been used in industrial application out there, quite successfully so, since a couple of years in actually making natural processes and making drugs in a sustainable way. And one example that was published in 2019 by the pharmaceutical company Merck was the process that led to the synthesis of this drug here called islatravir, which is an HIV drug. And what you can basically do is you can start with basic chemicals, use enzymes that you have engineered in the process, and put them all into one pot, stir, and in the end extract your final product from water. No organic solvents needed, no toxic heavy metal catalysts needed. So everything is sustainable as it is. So this is kind of, the process that was used for the synthesis of islatravir, but there are a lot of other application areas of enzymes. If you think of our transition to a sustainable bioeconomy, we'll need to be able to control those enzymes and need to be able to engineer them and program them in order to achieve a lot of those goals. And one of them, for example, is recycling precious materials, as for example plastics. And there a nice case study is here that in 2016, researchers from Japan went to this waste dump where we have a lot of plastic waste and simply scraped off some bacteria from there and found a bacterium called Ideonella sarkaniensis, and this bacterium actually grows on that PET and can metabolize this PET and use it as a carbon source to produce terephthalic acid and ethylene glycol that it can then actually grow on. And now what can be done after a lot of enzyme engineering, after a lot of process optimization, a French company called Carbios can actually use this enzyme in a hopefully soon scaled process. They plan to use this enzyme at 50,000 metric tons of PET a year in France to help us with our recycling problem. However, when it comes to finding those enzymes and making those success stories work, as for example PET degradation, as for example synthesizing small molecules, one key bottleneck is that we have to find those enzymes. We have to discover them from somewhere, right? We can't simply go to the environment and scrape off a plastic degrading bacteria from a waste dump all the time. That doesn't work for all applications, unfortunately. And then, once we found an enzyme, we also need to be able to engineer it. We need to be able to scale it at metric ton scale, and that means that we need to introduce mutations and control the way the enzyme works and program it. The problem is, however, that the space that we sample those enzymes from is incredibly vast. Right? So if we take a 100 amino acid protein, which is rather small actually, and we vary the sequence of this 100 amino acid protein to all possible combinations, we end up with a space called sequence space that is 10 to the power of 130 large. This is vastly more than the number of atoms that we have in the known universe. Meaning that not only can we not think about that space and imagine it, we can't even build it with all the atoms we have in the universe. And if we now want to traverse this space that we can show as a fitness landscape that you see here on the right, where a certain fitness for a given function is plotted on the z-axis, and sequence space is broken down to 2 dimensions, and peaks are now functional proteins in this space. We cannot navigate those landscapes yet. With AlphaFold, where Simon mentioned the Nobel Prize was given last week, we are very, very good at predicting structure now. But we are still not good at predicting function. We still don't know what kind of functional, or what function a given structure or a given sequence will have. So we still don't have the bioinformatics tools to actually navigate that space. And that means we actually need to sample it experimentally. And that means we need to go out there, sample natural diversity, test this natural diversity in the lab. That means, if we want to engineer a protein for a given function, we need to experimentally change a lot of positions in the protein, sample that in the lab, generate data, and find and identify mutations that are beneficial in the process that we call directed evolution. So we still, as experimental scientists, are useful in that kind of space, and we need to generate data. Maybe I hand over to you there.
Cool, okay. So where do I fit into this picture? My background is a theoretical physicist. Now I'm in the computer science department, and I build algorithms to help scientists like Max tackle these problems, specifically to design and engineer proteins. And engineering, to me, basically reads as optimization. So I have a starting point, and I just want to optimize it to be better. And design, to me, reads as sampling a starting point to begin with. And so really, if Max is the Batman, I'm the Robin to help accelerate what he does, so that we hopefully get solutions to various scientific problems sooner. And just to illustrate that again, Max has explained very nicely just how vast this sequence space is, and the classical way how we would do an engineering campaign would be through a technique called directed evolution. And probably many of you are familiar. The quick summary is just that you have iterative stages of mutagenesis. So you scramble your sequence a little bit, and then you subject it to selection pressure, and you keep on repeating rounds of mutagenesis and selection pressure in order to fish out the sequences that are ever better at what you're trying to do. And the challenge in that is often it's somewhat unpredictable. So it's a random process. Sometimes you end up somewhere where you want it to be, but sometimes you don't. And if you don't end up anywhere, you really don't have any insights as to why it didn't work, because you usually only see the positive results. So if you don't get anything, you don't learn anything that will help you in the future. And the other problem is it can be problematic to traverse sometimes gaps in that sequence space, because you need essentially a connected way of mutations that leads you from one variant to another. And so what the hope is with kind of artificial intelligence in that space is that we can start learning from protein sequence data that is related to function, and hopefully help with some of these issues that directed evolution is having in the engineering sense, namely speeding it up when something doesn't work, trying to understand why not, and when we encounter these kind of crevices in the fitness landscape, that we can hopefully jump over them and restart or rekindle a failed directed evolution campaign. And in the little picture on the right is just a slightly different analogy of a combination lock. It's kind of like if you imagine a typical protein of 300 amino acids, you've got a combination lock of 300 dials with 20 digits each, and now you want to find the code that cracks your particular problem, and that can be obviously very, very challenging if you ever try to pick a lock. Okay, so that's enough about us. Before we dive into the material, we just briefly wanted to also learn a little bit about you and to understand and tailor what we're going to talk about in the coming slides to your interests and your needs. And so I prepared a couple of questions just to get a quick overview, since I see we're more than maybe 20 or so. If you just raise your hand if you've ever trained a machine learning model. Okay, that's roughly 70%. Huh? Okay, nice. How many of you have used a machine learning model in your scientific workflow? Okay, so more people have trained a model than have used one. How many of you have pipetted an assay? Pipetted an assay or run an assay in a lab? Okay, also roughly 60%. So it's nice. We've got a mix of computational and wet lab experienced people. How many of you have engineered a protein? Okay. 3, 4. Okay, so we do actually have some people with protein engineering expertise. And how many of you, I guess now we've kind of explained it, but know what protein engineering is or feel like they roughly know what the goal of protein engineering is? Okay, nice. Very nice. And shall we ask for a few examples?
Maybe, yeah.
So we're thinking we just like ask for maybe 5 people or so. Just roughly, what's your name? What project are you working on? And what would you like to take away today kind of from this session, just to get a better understanding? And anyone here kind of willing to answer these 3 questions for us? Otherwise, I'm just going to... Yeah, wonderful.
Well, my name is Martinez. I'm from Mexico, and my project is basically focused on finding some biomarkers on the epilepsy and on electroencephalographic records with epilepsy. And what I'm trying to take away from this presentation is trying to see the EEG records as a sequence of protein, because as you said, there are like a lot of combinations of amino acids that will have like a specific function. Thank you. So in the same way, the signals on the brain are very different, and each waveform is specific for a certain biomarker of the epilepsy. So it's kind of a similar issue that I have. I want to try to find, or maybe, try to find which waveform is the most accurate for detecting epilepsy, and it's kind of like the same, trying to find which combination of amino acids gives you a specific function. So I'm trying to see the signal as an amino acid sequence. That's
okay, nice.
What my work focus on. Yeah.
Very nice. Thank you.
Yeah, thanks to you.
So biomedical data.
Yeah.
Do we have another person?
Hi, my name is Moises, but Moy for short. I'm from Mexico as well. We are from the same university.
Oh, nice.
My research focuses on implementing or using deep learning models to check or to study variation within the genome and try to figure out the grammar if we assume that there exists one, and to, let's say, predict based on variations within the genome if a person could be or could have any kind of disease.
Okay, nice.
Based on their genome.
Nice, very nice. So genomic language, essentially. Biomedical data, genomic language. Someone from that table? Do you have someone from that table? What are you working on? Oh, okay, okay.
Hi, I'm Mona. I'm going to start the project, so I haven't done anything related to that yet. So I'm going to start protein engineering using codon expansion. So I'm interested to see with adding different kind of chemical into the protein sequences, how would it change the protein folding and the functionality of the protein. And I'm also interested in seeing how we can work with different data that can apply in directed evolution as part of the protein engineering process. So yeah.
Great,
Awesome.
yeah.
Okay, so someone working on protein engineering. Shall we do that table?
Hi, I'm Jenny, but I'm not directly working with protein, but I'm working on mRNA display. And then finally we check if the mRNA we produce binds with the motifs that are corresponding to it. So it's partially RNA and then partially protein. So I'm just curious to know what can be, what information I can gain from here. So there's more spaces here, so I thought I'll join. So yeah.
Awesome.
Great.
Thank you. Wow, really diverse crowd. Very nice. Shall we take maybe one last from your table?
Hi, my name is Arianna, and I'm working mainly on metagenomic data, and especially in a cohort with some, let's say, workers in a sewage kind of environment, so to see if the impact of the pig microbiome is infecting also the human one. I'm not really sure why I'm here, like it's quite different. But I'm always, I'm a bioinformatician and I never worked on machine learning, so I'm always happy to learn something new and maybe to apply it to my research.
Nice, very cool.
Present and future.
Fantastic, thank you. Wow, okay, so super diverse crowd. Lots of expertise in the room, really exciting. I hope there'll be something for everyone, from microbiome to biomedical work. It'll be a little bit of a challenge, but we'll try, huh? So let's get started with a very brief whirlwind tour through AI, just so that we're using the same words in the same way, and that you understand how we are using these words when we use them. And then I'll get into data and AI specifically. We'll take a quick break and then actually the case study. So let's start with what is AI and do a little Venn diagram of buzzwords that are floating around in the space. So the most general word really is artificial intelligence, in a sense, or at least in the way it's used in the artificial intelligence community. And then artificial intelligence has a history that goes quite a while back, actually, right? Like it was used already in the 60s, 70s, and the first AI systems that started to be really powerful and showed effects for real-world problems came up maybe in the 80s, 90s, and those were mostly based on rules that were derived from human experts. And so the quintessential example is IBM's Deep Blue, so the chess engine. Maybe some of you know it. I see some people nodding. And so really that was derived by human experts who have chess expertise, scoring different positions, understanding, this position on the board will give me those opportunities in the future, this position on the board will give me that opportunities. And so really it was a system of rules that required a human to define what those rules are. And based on that, you could then have essentially a huge if-else tree. If this happens, then, and if this happens, and if this value is larger than that, then that will be smaller than that, then it's likely this option is your best pick. And so this already counts as artificial intelligence very widely, but people don't usually mean rule-based systems when they talk artificial intelligence today. And so the other buzzword that's thrown around a lot, besides AI, is machine learning, which is another subcategory of techniques in artificial intelligence. And machine learning really became popular kind of when people ran into the limitations of these rule-based systems. So kind of in the 90s, end of the 90s, people were thinking, oh, you know, if we just get human experts to explicitly write down all the rules, we'll have super generic general intelligence, and that can then solve all the tasks. So it's a problem of writing down all the rules. And very quickly it became apparent that it's just very hard to write down the rules. And even for tasks that we as humans are very proficient as, without, you know, any expert training, other than normal life experience, it's very hard to formalize them in rules. So if you ever, you know, see a cat and a dog, write down the rules that tell you what exactly makes this a cat or a dog, and you'll probably end up making a huge list and finding it very difficult to formalize. And so this other branch of artificial intelligence that's learning-based, machine learning, tries to, rather than write down rules, tries to just use data and extract patterns and statistics, use statistics essentially, on computers to extract patterns from data itself that you can then use to make decisions. And so early proponents are like decision trees that are now not, the if-else now is not branched based on what a human gives as a rule, but is extracted from data that is essentially trained, or a model that is trained on a data set. And then kind of around 2012, 2014 maybe was when it started to become really popular. We have this subfield of machine learning, which itself is a subfield of artificial intelligence, known as deep learning, where really the big kind of convergence was improvements in hardware, improvements in how do we initialize and deal with models that have many, many parameters, and just larger scale of data that is available and ease of accessing data that is available. Now the internet is obviously very, very present in people's lives and very easy to get large amounts of data for. And that's when you first saw, I think, was it 2012 or 2014, the kind of ImageNet moment of AI, when you first had classification models that performed really well in these dog-cat type classification problems. And the keyword really here in machine learning and deep learning, and maybe just an example of deep learning would be all sorts of neural networks would fall under the deep learning kind of taxonomy in this Venn diagram. And the key word on these two boxes on the right-hand side is the learning part, and that is the extracting patterns from data, extracting rules that we will use to make decisions based on data. And so on the bottom, you see this little slider from human designed rules to statistics. The more you go into that bottom right corner of the diagram, the more data you typically need in order to satisfy, essentially, specifying the parameters or tuning those parameters in order to represent the patterns you see in your data. And so really, the rest of this talk will focus on the learning part of AI, and we'll talk a little bit about pitfalls and how do we actually create and curate that data path that leads to learning in the machine learning and deep learning section of artificial intelligence in particular. Any questions or thoughts at this point? Okay, so far so good. Let's go into data and AI now that we've established that all the learning-based techniques fundamentally rely on data. And so this is this little Luigi here. And the reason I put Luigi here is because I think data is somewhat of an unloved hero of artificial intelligence. There's this really nice paper with a fantastic title from Google Research called "Everyone Wants to Do the Model Work, No One Wants to Do the Data Work." And it's a little bit cut off, but you can see here data largely determines the performance, fairness, robustness, scalability of AI systems. And this is a review that particularly talks about using AI in the context of society. But even if you look at, the cutting edge of the maybe more sexy AI that everyone talks about on the news, large language models, this is literally from the model architecture section of Llama 3. Llama 3 is the currently leading open source large language model, so it's about as powerful as GPT-4. And you can see that in the first sentence of the model section, they talk about how they use a standard transformer architecture from Vaswani 2017, and they don't deviate significantly with Llama to Llama 2 to Llama 3. And really, they attribute the performance gains primarily to improvements in data diversity, and of course, increasing training scale. And so they're very explicit that, that data part that you won't see a lot of papers at machine learning conferences about is actually what they believe is the strongest contributor to the performance of their models. And so that's why we want to talk a little bit about the data side of things. And so this was large language models and models in societal context. And I just put a paper up from today, or yesterday, on bioRxiv. There's also these protein language models. I think the same exists for genomic language models. And there's this very interesting paper from a Canadian group that came out this week, where they also reiterate this point by essentially, in their words, addressing data quality concerns. They managed to beat the performance or rival the performance of a very commonly used nowadays protein language model, ESM, ESM2, with a model that's about 10 times smaller and about 1,000 times faster, mostly by doing work on the data side of things, not really any fundamental changes in architecture, other than using flash attention, which is more GPU performant implementation. Okay, so hopefully that's enough motivation for why, even though data is maybe not the nicest topic to always talk about, why it's nonetheless very, very useful to do that. And so what are some considerations when creating data sets for AI in particular? Because the wonderful thing that distinguishes us from pure machine learning scientists in the sciences is that we can actually go out and create and design our own data sets. And so in many of our conversations with Max, the question was, well, if I do need a data set, what should I look for? How should I design it in order to make it as amenable as possible to gain as much as I can from AI models? And so the very first question that always comes up is scale, right? Just how much data do I need for what model? And I think there's really no one-size-fits-all answer for this, because what matters is not really the scale or just the count of examples, but what matters more is the count of meaningfully different examples. So how much of the actual diversity of your space that you are trying to model do you capture with your data set? If you want to model the particular enzyme that we'll talk about in the case study, if you can model locally the interactions, there is a different answer than if you want to model enzyme function at large for all kinds of functions. And there's probably also, again, a very different answer for if you want to model protein stability. And the requirement there is probably lower than if you wanted to model enzymatic functions. And so this is this example. What you put into your model, probably many of you are familiar with this XKCD comic, is roughly what you get out. So the quality of the data and the amount is roughly what you get out. And so really, when you think scale, always think as well diversity. Can you have enough semantically different clusters that you can capture? And I think especially in bioinformatics, there's been fantastic work on understanding how to cluster similar examples and understanding diversity within those. And just to give a specific example of where this was used in the biological sciences, this is a paper from Charlotte Dean's group at Oxford on canonical shapes of complementarity determining regions of antibody loops. So this is the main region that contributes to binding in antibody-antigen binding. And what they were doing was they were holding out some training. So they first clustered the data set according to so-called what they call canonical shapes. So they clustered on a structural similarity basis the CDR loop regions in their data set, and then specifically held out a couple of clusters from training that were structurally significantly different. And the question they asked was, if we just scale the number of data points in our training data set, do we ever get anything from a different cluster? So do we ever improve performance on those clusters that are structurally different, then have a different canonical form versus what we trained on? And the answer was actually no. They never found anything that fit any canonical cluster that was not in the dataset. And so that again tells you that these models are not magic black boxes. They very much depend on what you give them. What they did find, however, was that when they added only a single or two examples of a different canonical form, they would start seeing more examples of that canonical form and better performance on that other canonical form in their validation. And so to me, this is kind of the digest of this research is a little bit that if you can, spend a little bit more time getting an extra diverse data point, that you don't necessarily need to have a huge amount of those. And it can already significantly help you if you do these type of generative tasks. So if you try to generate antibody CDR type regions. And this is just another example, again, a real-world example of a study that underlines how diversity is important. This is from a company called Basecamp Research, and what they did was they go out and they just sequence metagenomic data all across the world, and then they use that data to retrain a version, or not even retrain actually, to use the existing version of AlphaFold and just change the way the MSAs are computed by plugging in their databases, which just have many more representatives. And they got significant improvements in the performance of protein structure prediction by providing data that was more diverse. And so that gave the model access to some of these statistics on sequences that you wouldn't find in UniProt. So much on data and scale, and depending on what you're trying to do, and if you have the time to obtain extra data, you might want to spend it specifically on getting diverse data, meaningfully different data in your assay. The next step the next kind of element in an incomplete list of what makes data sets useful for AI that I wanted to talk about is integrability. So how can we integrate data from different sources? And one example here that's very challenging in the space of enzyme engineering is that we use this word fitness or function, and fitness or function can mean thousands of different things. And it can also, for example, there is this data set, Protein Gym, and they've now massively improved it, so credits to them for really going and drilling into that complexity. When they started out, you had, they went over, as machine learning scientists, so they went over hundreds of studies of deep mutational scanning data sets, and they would find, the data sets and the fitness values that came from these studies and collate them in a benchmark in order to allow us to track how we're doing in modeling different tasks. The only problem with that was that some of these data sets corresponded to enzyme function. In others, fitness corresponded to stability. And yet others, the fitness value corresponded to viral escape. And so what fitness means is fundamentally different for many of those. And so it's unlikely that you would expect one model to, when you train it on generic fitness and then ask it, what would be the fitness, to benefit from learning whether a particular sequence is better at viral escape, so probably at varying a lot from being very stable or from being very or highly catalytically active for this particular substrate. And so it really, and kind of in the next iterations of Protein Gym, the authors actually started subcategorizing into similar types of function, similar types of tasks, and saw improvements in sharing data across different modalities through having meaningful subcategorizations. And so it really pays to understand which data sets can I integrate, when can I do that, are the tasks meaningfully similar, and am I expecting to be able to learn something from this particular data that is useful for the task I'm tackling. And when integrating data sets, one thing that's super important is consistency. And so when you scrape your data sets from the internet, as me and others would do that mostly come from not having a wet lab at our disposal, one thing we need to be really careful of is what is the dynamic range that was used in that assay, right? We'll hear that later in the case study, but in the fitness assay that Max was using, we have a different resolution for variants that are around as good as wild type, variants that are, definitely better than wild type, say 3-4 times as good, and variants that are stellar, 100 times as good as wild type. What we would be able to distinguish based on that assay would be, is it around wild type or is it better than wild type? What we would not be able to distinguish, is it a great variant or is it a stellar variant? And so depending on what your data can tell you, and that really isn't reflected always in the numerical values, you have to treat it differently in the modeling. And we'll hear more about that in the case study. And that's relating to the dynamic range of your samples. Beyond dynamic range, it's also really important to understand what does it mean when you couldn't get a value. In our case, when you couldn't get a value, it's a deep mutational scanning assay. Again, we'll go into the details a little bit later. It often means that that example just wasn't sampled in the sorting step. And so we really can't say anything about it rather than saying that it was a bad example or there was something faulty with it. Another point that's really important about consistency is can we quantify the uncertainty between different data points? And really understanding what can we learn. In the example of quantifying the uncertainty, in our case we have quite a bit of noise in the assay. And so what we can say is if a variant is meaningfully better than another, if they're separated by their relative kind of confidence intervals. But what we cannot say is we can not order the 10,000 variants individually on just which one is better than the other, because that's not within the resolution of the assay that was performed. And so it really pays to understand these. And if you have AI collaborators, or if you're doing the AI work yourself, if you're doing the AI work yourself and you're doing the experimental work yourself, you're really benefiting because you know the ins and outs of what assay you ran. If you're collaborating with someone who's running the assays, or if vice versa, you're running the assays and collaborating with someone who does the modeling bit, it really pays to sit down and discuss in detail where do you expect the error sources to be, what dynamic range, what resolution, what do different values actually tell you, and how much can you rely on them. And if you want your data to be used by others, it's super important to have it accessible. I mean, the PDB, etc. There's wonderful places and precedents on how to do that. But what's also really important is, are they self-describing? Are there metadata that tell you more about the data itself? And that's really, really useful if you want people to use your data meaningfully, because if you have to collect many data sets across a diverse range of, say, deep mutational scanning studies, like in Protein Gym, there's over 100 different deep mutational scanning studies that go in there. It is a lot of work to read and understand 100 papers, especially if you're a computational scientist and you don't know the details that go into these particular assays that are run for these deep mutational scanning data sets. And so having self-described data via metadata is absolutely crucial if we want to help each other in the field. And then versioning is another thing to think about. How are you going to have future versions of your data, and how are you going to release those versions of the data? So this is if you want to share it with the community at large, which obviously we encourage you to do. And then the last thing on my incomplete list of, or incomplete data wish list or considerations list is bias. How do I control for extraneous effects, influences, spurious correlations? And here again, an example. This is also taken from the case study that we'll look into in a little bit later. But when you do protein engineering, most mutants are bad. They just don't work. And there's a few mutants that are really, really good. And when you evaluate how well you're doing on a task, some metrics are sensitive to, or some performance metrics are sensitive to what is the majority class. And so in particular, one metric that is used very often in protein engineering is Spearman correlation, because if you have a good Spearman correlation, that means that you're good at ranking, relatively, which variant is better than which other variant. And so the higher, if you had a Spearman correlation of 1, that would mean you would perfectly rank which one is the best, which one's the worst, and anywhere in between. If you had a Spearman correlation of 0, obviously you would have absolutely no clue and you would not get any single pairing right. But Spearman correlation is not actually what we care about in protein engineering, because we don't care about telling or understanding just how bad a bad variant is. We really only care about saying which ones are the great variants, and of the great variants, maybe rank ordering those. And so the kind of slightly shocking thing, though, when you use Spearman with a data set like the one we had, was we had roughly 10,000 mutants. And so this is just 4 synthetic data sets that I generated to show you how Spearman behaves with an imbalanced data set. And so at the top left, you see the ideal case that you would hope you would be in, right? You have 2 clusters. This is the cluster of mutants that are generally bad, and at the top right is the cluster of mutants that are generally good. And here your model is great at ordering both the bad and the good data set and within each other. And so this is the great case, and you see the Spearman on the low fitness is great, the Spearman on the high fitness is great. If you can't read it, it's both around 0.9, and then the combined Spearman is 0.9 as well. Binary Spearman, so whether you're in this cluster or that cluster, 0.6. What happens if we're in a still good case for protein engineering? So we still manage to rank all the good variants well and better than the bad variants, but we have no clue about the bad variants. Well, now we have a 0 Spearman on the bad variants and a combined Spearman of 0.3, or 0.37, pretty low for machine learning purposes. For biology, 0.3, 0.4 is actually not unheard of. But we're really good on the high fitness variants, so 0.9. And so this would be just as good as this one for actual protein engineering. But Spearman here is just, significantly worse, from 0.9 down to 0.4. On the other hand, what if we were really good at, ordering just how bad a terrible variant is, but had no clue which ones are good, which would be, here we're still able to do the binary task of whether it's in the good batch or in the bad batch, but we're not actually able to say whether something's just as good as wild type or great. And then we still get a Spearman of 0.9. And so you can just see how the Spearman is massively influenced by whichever class you have more data points on. The big difference is that we have 7,000 bad data points and only 1,000 good ones. And so this is just a cautionary tale that how you evaluate on whether you make progress is extremely important. And it's very important for whoever does the modeling to also understand what is the actual ultimate task, or for everyone on the team, really. And that was one thing that we also had learned in kind of the collaboration. I was originally, looking after machine learning metrics. I was like, okay, great, let's have a great Spearman, let's have a great binary classification metric, etc. And then was brought down to the ground by Max, explaining to me, what the actual goal is that we want to achieve in protein engineering, and that is not actually reflected by anything we did in the machine learning community at the time. All right, so that was a quick list of considerations and a wish list on data sets. Any questions or thoughts or additions at this point from any of you, or you, Max, or Matt? All right, sounds good. Then on that bias bit, just before we go into the coffee break, I want to spend a little bit more on the bias bit to hone into why it's so important to look at your data in more detail, and kind of what are some of the fundamental pitfalls you run into specifically with these learning-based models. So this is the same boxes that we had before. This is from a review we wrote recently on AI in protein engineering. So this is a summary of all studies that were done in protein engineering using AI in the wet lab. So if you want more details specifically on the protein engineering side of things, I encourage you to check that one out. I think we'll make the slides available after as well, and you'll find the sources on the slides. But to understand one of the common pitfalls, let's go back to images because it just speaks to humans very easily. And let's build a classifier between cows and penguins. And so I just went on Google and I got pictures of cows. And so you see a lot of pictures on cows. So I put that into the cow data set. I did the same. I make a data set of penguins, and I put it into my penguin data set. And now I want to train a classifier to tell me whether it's a cow or a penguin that I'm seeing on the picture. Do you notice the issue with my data set? Exactly. So this is a cow, and this is a penguin, and this is a cow, and this is a penguin. And so the issue really is that there was a spurious correlation, namely the value, the green value or white value of my image in that data set. And the model, rather than figuring out the complex task of understanding what a cow is and what a penguin is, just figures out, is there more green or white? So it was a cow or it was a penguin, because that allows it to do really well on the data set that they're using to evaluate. And so this is a story of spurious correlations and heuristics, and how these models, these learning-based models, so everything we saw on the original bottom right corner in the taxonomy I had, or the Venn diagram, are extremely prone to exactly this type of error. And so this is almost cartoon-like, right? So let me give you another example where this happens not only in artificial intelligence and these simple systems, but this is now an example of biological intelligence around 1900. What you see here is a horse called Clever Hans. And Hans was called clever because it was apparently ostensibly able to do mathematics. And so you see a group of psychologists and pop psychologists gathered around that horse in Germany at the time. And it was, people were studying the preconception or the preconcepts of mathematical reasoning in animals. Obviously, the human's always at the top, where we always put ourselves. And then you've got, maybe horses, maybe mouses have slightly fewer mathematical concepts, etc. was the thinking at the time. And so why was this? They would actually write some arithmetic equations on a blackboard, and they would then have the horse point to, what the correct answer was. And the horse was phenomenally good at doing basic arithmetic, addition, etc. And what they found was when they removed the horsemen from the bystanders that were observing, the horse suddenly couldn't do any maths or arithmetic at all anymore. And so what the horse was extremely good was at reading the very subtle cues that the horseman had with the right answer, because the horseman could actually do arithmetic. And so the horseman would spend, maybe a split second more with the eyeballs on the correct answer. And the horse is, not trained for arithmetic, but it's very, very good at, emotionally understanding the thoughts and feelings and processes that might go on in the horseman. And so it picks up on these heuristics and can solve tasks that might look like it's doing something else, even though it's actually doing maybe more emotional intelligence rather than mathematical intelligence. And so this was the example already for biological intelligence. The same thing happened for artificial intelligence with language models in 2019. So this is BERT and the world in 2019. BERT was one of the first large language models that was actually used. It started to make its way into Google search in 2019 and was one of the precursors of the large language models we're seeing today. And so the Clever Hans story in AI style, this was a research group that looked at whether BERT can actually do reasoning, kind of the way it was trained. And what they trained it on was these snippets of data sets of questions and answers where you were supposed to do a few bits of reasoning in order to understand what the correct answer is. And so the question here is kind of like, what was the name of the quarterback who was 38 in this and that Super Bowl? And then based on that paragraph, you should be able to figure it out. Now, that BERT model did phenomenally well on this benchmark. So this is a benchmark of like 20,000 such questions. They evaluated BERT on how well it did. It completely blew out of the water any model that existed to that date. And so at that time, they could have written up and had it published in a nice machine learning conference proceeding and move on with their lives. Luckily, that's not what they did. They actually started to try and understand, why does the model perform well? Why does it perform so much better than many of the other precursor models? And what they found out was when they added an extra sentence at the end of many of these paragraphs of the who is type questions, the model would just return whichever named entity was the last named entity in that paragraph. And why did it do that? Because in the data set, you had this spurious correlation that many of the questions that were paired with who is type questions happened to have the person who is the answer at the end of the paragraph, or basically it was just the last named entity of the paragraph. And so it's how that data set was constructed that determined what the model learned. And the model was like, oh, I can do this task by just telling you the last name of whatever paragraph you're using, rather than really having to, quote unquote, think about what the problem is. And so when they added just a random name at the end, where you would now really need to do reasoning to kind of figure out which of the two, or really need to do reasoning, at least some sort of inference to figure out which of the two named entities, and you break that spurious correlation, they found that the model was actually not better than any of the other models they had previously anymore. And so it was very good at this kind of named entity recognition, but a lot of the performance went away when they corrected for spurious biases in the data sets. And that really tells us that when we want to understand how good we're doing, we need to make sure that we kind of cleanse our data sets of accidental spurious heuristics, and that we also need to be really careful and take every result that we get from these data sets with a grain of salt, and understand whether it might be due to some heuristic in the data set or due to the capabilities of the model that we're actually making progress. And so this is where I guess experimental design comes in a lot. And so just the bottom line, they have a wonderful sentence in that paper that statistical learners, so anything in that learning box, are prone to adopting shallow heuristics instead of learning underlying generalizations. And I like to reformulate it as Murphy's law for deep learning and shallow heuristics. If a model can solve a task with given data with a shallow heuristic, it will. And how do you avoid this? Well, mainly the tunable knobs are adaptations in the data, adaptations in the model itself. So there are some techniques you can do to avoid certain types of overfitting, and adaptations in the way you phrase the task and the way you measure performance. And so there's no silver bullet, but just a few points on how you can address some of these issues with spurious correlations. Of course, more and better quality data always helps. Incorporating model confidences helps, because what that allows you to do is to understand whether the model is, confidently wrong in what it predicts, or just doesn't know, but it has to predict something because you're tasking it to give out the value. And so whether you shouldn't rely on it. And I think the best example in the sciences is the PLDDT metric that AlphaFold uses in order to tell you how confident it is at various ranges. And I think one of the reasons AlphaFold has become so much use is actually because people can somewhat understand whether they should rely on whatever AlphaFold says, or whether they AlphaFold just gives you some numbers because it has to, but really has no clue. And so I think in the sciences, incorporating model confidences and calibrating confidences is very, very powerful. There is a whole section of machine learning that deals with calibrating confidences and calibrating predictions, and it's called conformal prediction. Highly recommend checking that out if that's of interest to you. Another point is ensuring robust training and evaluation protocols. So what do I mean by that? I really mean thinking deeply about what the scientific question we'd like to answer is. And so how do we split our data into, what is the test data set, the validation data set, what goes into the training data set? Do we actually meaningfully split away clusters? Do we have the same cluster represented in a training set and a test set? Well, then we're asking a question of, if we had some examples of a cluster in our training data set, would we be able to generalize to other examples in a similar cluster? What we would not be able to answer based on that evaluation is, would we be able to generalize to new clusters? And so we would need to keep a cluster entirely separate from the training data set if we want to answer that question. So really, depending on, and sometimes we're okay with generalizing within a cluster. There's nothing wrong with generalizing within a cluster locally. It's just that we need to be aware that that's the scientific question we're asking based on the way we're splitting our data. And so different splits, different similarities in the case of biomolecular modeling really determine what scientific question you're asking of your model. Information leakage is another thing to be aware of. Again, that goes a lot with kind of splitting your data. Augmentations and invariances allow you to kind of better capture the covered space. A lot of that is in the field of geometric deep learning. So maybe you have crystal structures. It doesn't matter whether you look at your crystal structure from down here or from over here. It is invariant to global rotations and translations. And so your model should also accordingly be invariant to that. And then rigorous cross-validation as really one of the only ways to make sure we're actually having signal. Something else that's useful to understand whether we've potentially accidentally run into a shallow heuristic or not, and to try to mitigate that a little bit, is to use models, to always use a baseline, of course, because if we're not doing meaningfully better than the baseline, there's probably a simple heuristic that can already be modeled by a baseline that allows us to perform well on that task. And when you do choose baselines, or when you do choose kind of a progression of increasingly maybe more complex or parameter-heavy models, try to make sure that they are interpretable. And I mean, you've had a whole section on explainable AI in the morning. This is one explainability technique. I guess there's two main kind of branches. There is the retrospective interpretability, and then there's the prospective. Do I know how my model would behave if I changed this or that part of the architecture of the data set? SHAP values are a retrospective way of analyzing, so they allow you to tell apart kind of the contributions of different components during training. We'll again hear in the case study how we used that and understood that in our particular protein engineering example. But that can be really helpful for debugging as well. So for understanding, which data points or which features of which data points really seem to help with performance, which things are classified confidently, which things are not classified confidently, where do I need to get more data? So these models can also be helpful as a tool for guiding exploration. And then, yeah, I guess that goes into the next point, looking at a curated set of examples, really having, I think, a sharded list of difficulties is very useful. So in the development of AlphaFold, when you talk to the AlphaFold developers, and I've recently also been involved with an AlphaFold free replication effort, one thing that we're always using is data sets sharded by difficulty. So we have one data set that is easy, one data set that we deem more medium, and then one data set, or test set I should say, that we deem as very hard. And so if we do model changes, there might be no signal on the hard data set, just because it doesn't make enough of a difference to really change one of the really difficult examples. But we might see in the kind of closer resolution of the medium difficulty data set and a small data set that there is a difference. But then which kind of architectural decisions should really stay and which ones shouldn't, those should probably be the ones that make a jump on the hard data set. And so sharding your evaluation by difficulty can be really useful to kind of differentiate out that resolution of whether you're making progress or whether it's just noise in your validation data set that now gets you better. And then I guess as a last point, in machine learning, we love having benchmarks. Always be aware of p-hacking and benchmarks. If you have 1,000 people submitting models to benchmarks, at some point one of the models will randomly perform well, and then we'll latch onto that, and it'll be a runaway effect. And so that's why benchmarks always need to be updated. Benchmarks, just as a model, a benchmark needs to be updated. And I mean, CASP is a phenomenal example of how they did this in the past. I think we can all learn from how the CAS competition worked. Sorry?
Like benchmarks need to be updated, but in respect to what?
In respect to essentially what data points you're using. And so just to give the example of CASP, what they would do is every, I think 2 years, are they in a 2-year or on a 1-year cycle? For sake of argument, let's just say it's a 1-year cycle. It might be a 2-year cycle. They would completely change the competition, the data points that they would evaluate on, and they would not show it to anyone on the world, essentially. They would have a committee that would have access to the crystal structures of that data set. And so people would be allowed to train models based on any publicly available data before then. But then the evaluation would happen on that test set, which changes every year, in order to really understand our progress in protein structure prediction. And that's in contrast to many of the other benchmarks that you see in the machine learning world. So MNIST is a very popular benchmark that people used in maybe 2014. Nowadays, it's completely saturated. It's handwritten digits, and so that hasn't changed, right? ImageNet hasn't really changed. Many of the benchmarks that are used in the machine learning communities are not updating what they're evaluating or the examples that they're evaluating, and then you expose yourself to p-hacking by just trying and submitting more and more models over time. And so, yeah, benchmarks, as with models, ought to be updated. All right. Perfect. Any questions, thoughts?
I jumped the gun then. There was one question on the chat that did pop up as you were speaking. I think it popped up during the explanation of BERT, and how that it was picking up on that correlation that the name was normally coming at the end of the paragraph.
Oh, okay, yeah.
So the question was raised by Manuel. Could this be solved by analysing the embeddings from each LLM and detecting these correlations? Correct me if I'm wrong, Manuel, on the chat.
Yeah. I think that would be one way of trying to understand and debug those. I think with LLMs, yeah, you do have the attention maps, which go some way towards explainability. It's not explainability in kind of a white box sense that you would know what would happen if you changed it. You actually have to try it out. But I think the way we're dealing with these models actually is very similar to the way we're dealing with biological systems. So you throw in a different input, you probe what happens, and you try to figure out kind of what goes on under the hood, essentially.
So taking a more experimental approach, I guess.
Exactly, taking a very experimental approach. So almost like a biology approach to that, system. I mean, biology has made remarkable progress this way. I think even here it can be very helpful as well.
I like it. Very applicable. Very applicable from wet lab to dry lab.
Exactly.
In this
yeah,
sense
exactly.
As well.
I think there's a lot from us
bridging the gap
as dry lab people to learn from wet lab scientists as well on how you interrogate your systems.
Thank you.
What was really exciting here is just that they, in the end, went and actually corrected this bias in the data sets, and then really saw what that had as an effect in the downstream predictions. And so that was really exciting. So obviously, if you have text, it's a little bit easier to do. How do you correct the data point that you took from an essay? Well, I guess it's very difficult. Maybe you can vary one of the, so if you think there is a spurious correlation, so for example, spurious correlation in enzyme catalysis could be temperature, right? Maybe you did all your measurements in summer and it was a warm room, and so purely by virtue of high temperature, you had faster diffusion and faster reaction rates. Maybe you do your next experiment, at a controlled temperature, rather than having one data point in winter, one in summer, or something like that. So if you can think of likely spurious correlations, or if you can detect kind of some surprising results, kind of go and double click on those and see what might be a spurious correlation that the model could have picked up on.
Thank you.
Thank you for the question. Any other questions or thoughts at this point? Because then I think we're ready for a quick coffee tea break before we go into the actual case study. Yeah, sounds good. Let's do 10 minutes.
All right, does everybody hear me? Awesome, perfect. Yeah, so that's the next part of the session, now that Simon has given you such a wonderful introduction into kind of what you need to take care of when it comes to the AI model side. I'll go through a case study, and this is very, very heavily in the kind of protein engineering, enzyme engineering field, where Simon and me have collaborated. And we also brought a third collaborator, Matt Penner, who is also a member of the Hohlfelder lab, and who has collaborated with us on kind of another study, which he'll talk about kind of quickly in the end, just to give you a more diverse set of examples to kind of sample from. So what we have discussed before is we want to use enzymes for sustainable application, right? So we want to use those enzymes in synthesizing small molecule drugs, in recycling plastics, for example, or maybe in the future even to pull out CO2 from the atmosphere to mitigate climate change. And huge issue is that we have a lot of those enzymes in nature, but they do other things in nature, and they're not necessarily at the right setting yet to be actually ready to use in industrial application at scale. So we usually need to first find them and then engineer them. And we talked about this already. This sequence space that we need to sample those enzymes from is incredibly vast. So what do we do in our lab, where Matt is also part of, is we ramp up the speed of experimentation. And that very much goes back to what Simon mentioned before, as kind of one key thing that we want to supply in kind of AI models is amounts of data and scale, right? So what we do in the Hohlfelder lab in the Department of Biochemistry is we ramp down the volume of experimentation from the milliliter format to the microliter format, maybe in plates, right, where we can screen maybe 200 or like 500 variants a day, down to the picoliter scale, where we can go into droplets via droplet macrofluidics and ramp up our throughput of experimentation actually to the billions even. So how do we do that? How do we scale down the volume and thereby scale up the amount of data that we can generate? We can basically mimic what we have in the macro scale at the micro scale by making tiny little droplets. And the way we do this is we use those tiny microfluidic chips, which are in the micrometer scale, where we take, for example, substrate solution and individual cells, and we can encapsulate them in those tiny little droplets at throughputs of 2000 hertz. So basically 2000 of those droplets are generated per second. What we can also do is we can manipulate those droplets. We can add different volumes, we can remove different volumes, and we can do that in the process that we call pico-injection, which is basically like a micropipetting step, right? This would be at a speed of around 500 hertz, which is 500 additions of volume to different droplets per second. Yeah? And then in the end, what we can do is we can interrogate those droplets using those tiny optical fibers that you see here, and then sort individual droplets, upon, for example, they reach a certain threshold in some, absorbent or fluorescent assay that we have. We can then sort them out here at a speed of around 200 per second, right? So basically what this gives us is a tiny little plate reader where we can then interrogate those droplets individually at very, very high speeds. So traditionally, droplet microfluidics has been used in enzyme engineering to screen vast amounts of different enzymes in a format that we call a library. For example, from some kind of metagenome out in the environment, or for example, an artificially generated library in the lab, to interrogate this small portion here of the fitness landscape, that you see here, and to find hits. Right? To find an enzyme that does a certain job, or to find a mutant of that enzyme that is improving in its activity, for example, in a bioreactor or something. So what we've been doing in collaboration with Simon is we have transformed the way we think about microfluidics as a hit generation device and started to think about it as a data generation device. Because every time we sort a droplet here in this microfluidic chip, we make a sorting decision. And we wanted to transform the way we think about that data from simply sampling those hits, but to sample individual data points on the way. And the way we do this is we match the screening power of the microdroplets with the reading power of next generation sequencing. So what we can do is we can sample the input library and the output library after sorting with next generation sequencing, and then count individual members in our input distribution and in our output distribution in a process we call deep mutational scanning. And this now basically gives us a fitness value for each individual library member and tells us roughly, is this library member fitter, more fit than the wild type, or less fit than the wild type, and gives us also some kind of quantifiable amount of how fit this variant gets. Yeah. So basically, by combining microfluidics with next generation sequencing, what we can do is we can transform microfluidics into a data generation device and generate large-scale sequence function data. And this now, hopefully, with the help of AI, or in our thinking, allows us to go and extrapolate to a much broader space of the fitness landscape, yeah? And to maybe even find those like larger peaks on the fitness landscape much faster than what we could have done without the data. And I hope I can convince you, over the course of kind of this case study, that we've been able to do that. So what is the thing that we are trying to do? What we usually do in biocatalysis in an industrial setting is we get a certain molecule, this molecule here, for example, and people tell us, synthesize this molecule, right? And synthesize it in the most sustainable way possible, so use an enzyme. So then we go and we go into a database, or we screen like thousands of different enzymes that we find in nature, and we find a starting point. And we find something that has some kind of activity for our substrates, but this activity usually is not satisfying. So we need to go and we need to engineer the enzyme in order to get an activity that we then actually can use in an industrial setting. And this is done in a process we call directed evolution. For this, the Nobel Prize was awarded a couple of years ago. And this is basically an iterative cycle of mutagenesis, where we introduce mutations. We then find the improving mutation, and then we introduce mutations again and screen again and go into a cycle until we have satisfying activity. And this is basically this walk on a fitness landscape that gets us from a wild type activity here to something that we can then call a hit, and that we then can use in an industrial setting. However, as you can imagine, this walk on a fitness landscape can be very long. We cannot estimate how long this walk is going to take, and success is unpredictable. We don't know if we'll actually end up with an enzyme that is good enough to do its job. So this is very, very unsatisfying for industry. And industry usually tells us this is not good enough. This is really bottlenecking our use of enzymes in industrial application. So what we try to do is we try to use this technique, right, and map out the local fitness landscape around the wild type, and then ask, okay, can we use this data to estimate how well this engineering campaign is going to work, and can we use the data to extrapolate towards this higher point in fitness? And one kind of, criterion on the data was that we wanted to have this data tell us something about epistasis. So what does epistasis mean? What is hampering engineering campaigns a lot of times is that single mutations that we acquire in the engineering campaign are simply not additive with each other. They are usually worse than additive in something that we call negative epistasis, and this is hampering those directed evolution campaigns and creates those cliffs on the fitness landscape, right? So we wanted to have this feature in our data to help avoid the protein engineer falling into those cliffs and kind of avoid negative epistasis. Okay, so what was the actual reaction that we were looking at? This is a reaction we call reductive amination, where we want to bring a ketone here together with an amine to form a so-called imine, and this imine is then reduced by a so-called imine reductase, or short IRED, using NADPH as hydride donor, as cofactor, to form this amine bond. And why is this important? Actually, 35% of all pharmaceuticals out there have those chiral amine bonds, and it's incredibly hard to make them chemically. So making them chemically involves a lot of toxic solvents, toxic hydride donors, as for example sodium cyanoborhydride. So it's incredibly hard and toxic for the environment to make them chemically. So let's make them biocatalytically and use our technique to inform the engineering of those IREDs. How did we do that? We first needed to generate an assay in droplets, right? This is not trivial. We need to sit down and think, how can we generate a colorimetric readout from our reaction? So we coupled this with this reporter dye here, and we used all kind of steps in the repertoire that we have in microfluidics, droplet generation, pico-injection, and then sorting to set up a microfluidics assay. But much more importantly, how do we generate data from that? And very importantly, again, how do we generate data that includes this feature of epistasis that we care so much about to inform protein engineering? So therefore, we invented this workflow that we call long-read deep mutational scanning. And this involves different deep sequencing techniques. And one very, very important feature of short-read Illumina sequencing, as the name says, right, is that we can only go up to around 600 base pairs in read length. That means we lose out on all those long-range epistatic interactions that we find in a lot of genes if we do random mutagenesis, right? So if we have one mutation in the beginning of the gene and one mutation in the end of the gene, and we do a pooled screen, right, we lose the information that those two mutations are connected with each other if we only read with short reads. Yeah? So what we did is we combined Illumina sequencing with a technique called Oxford nanopore sequencing, which is a long-read sequencing technique. In this process called long-read deep mutational scanning. And what we did is we had this random mutagenesis library, single mutations, double mutations, triple mutations. We barcode each individual member with so-called unique molecular identifiers, UMIs. And those UMIs then tag each individual variant. And we can then say, okay, this variant belongs to this UMI and so on. And then we sequence this library using Oxford nanopore sequencing. As many of you might know, Oxford nanopore sequencing has a very high error rate. So what we needed to do is we need to polish out those errors by averaging out in our clusters. And we can then generate a dictionary where we link up our unique molecular identifier with our mutations, right? And then we have saved, okay, we know this UMI is this mutation. Then we can go back and we can take the same library and screen this library using microfluidics. Yeah? And then we afterwards count using simple Illumina next generation sequencing only the individual UMIs. And now we have the fitness information on the UMIs and we have the mutational information on the UMIs from nanopore, and we can then combine this data to actually generate this position dependent data that also gives us some evidence on epistasis. So what we then did, and this was also very important to convince Simon to collaborate with us, we went back to the lab and asked actually how good is this fitness information, right? So how noisy is this data actually? So we picked 14 individual members of the library, randomly distributed, and we correlated what we see in a plate-based assay, which is very reliable, to what we see in our data. And we drew up this correlation here, which is kind of reasonable. So this correlation works and tells us there's some quantitative information in there. But one caveat, as Simon already mentioned, is that the dynamic range of this assay starts to level off at some point here in the upper range. And then it's very hard for us to differentiate between very good members of the library and intermediate good members of the library. And what we can also see is, we here have a log scale in fitness, and we also have here a log scale in our, readout from the plate assay. So if you can imagine that it's very hard for us to rank actually individual members here. Yeah. So this data definitely is more noisy than the data Simon was used to in working before, right? So we really had to spend a couple of months, with Simon trying out different kind of model types until we found found out, oh, the data is actually much too noisy for a lot of those model types. So communicating your noise level is a very, very important thing when it comes to collaborating between, the experimental side and the AI side of things. So at that point, do you have any questions or thoughts kind of on our experimental workflow? Nothing, all right, over there.
The slides before, the sample size is 14.
Here, yeah.
So with the sample size being 14, how does it work with machine learning? Because I assume machine learning has to be large data set.
Sorry, then there was probably a misunderstanding actually. Actually, just our test set where we tested the accuracy of the data was 4. Actually, the data size, and I didn't say that, I think is 17,000. So we had 17,000 data points that we generated on the fitness, and 14 was only the number of data points that we used to evaluate if our data is actually, or how noisy our data actually is.
It's like a quality assurance plot, right?
Exactly.
QA,
No worries. All right, then let's dive into what this data can tell us, right? So as I said, we had 17,000 data points on fitness of individual variants. Yeah, over here, no worries.
Bit of time to shape. So how many points did you use for your test and your training data set?
Maybe you can directly answer that, Simon.
You'll see a little bit later. We actually use different amounts to also ask questions kind of differentially. How much would adding more data actually improve model performance? But ultimately is roughly I think 80-20 split for the end. Yeah, and then very importantly, your test set is kept entirely separate from your validation set, which is used for your understanding which model choices to make, and your training set, which is actually going into training the model. And so the test set is never touched during model architectural changes.
The training and validation?
Not purely randomly, actually, and that goes into kind of the things that Max was mentioning about error rates and understanding the confidence. So we cared much more about the positive representatives, and we cared particularly about the understanding those that are high confidence, essentially. And so what I did was I did a biasing of whatever is high confidence and to have an overrepresentation of the ones we care about, essentially, in the data sets we use to assess how well the models are doing. So in the test data set and validation data set. So that meant we had less to train on of those, but we had clearer visibility on how well we're doing at different stages.
Maybe actually before we dive deeper into the AI, because we will have a couple of points on the models that we used and so on even later. Just a very quick point on how or what we could actually do with that data, right? So this data really enabled us to profile that enzyme, get a mutational profile for our enzyme. And the first thing that you can look at here is if we map the average fitness that we find per position, which is something we call mutability, right? How expensive is it to put a mutation into a certain position in the enzyme? You can map that onto the protein, and you can find very, very interesting things. So if you look, for example, here at the substrate binding pocket, you find that nearly all positions around the substrates are, mutations that are very, very detrimental to fitness. However, there's this one very counterintuitive example here at the bottom of the binding pocket, 3 and 241, where basically everything we put in at this position is actually beneficial. So we can really find hotspots at the binding interface with the substrate, which can help us to engineer the substrate specificity of this enzyme, which is very, very useful for us as protein engineers. Another thing is that we can look beyond kind of what we find in the first shell of interaction with the substrate, kind of beyond more broadly on the enzyme. And we can find, for example, those, we have this domain swap dimer as our protein. We can find this line of hotspots linking the two the two active sites, which could tell us something about cooperativity, for example. So what I wanted to tell you here is that we can also learn something about the enzyme using those profiles and learn something about maybe even the evolutionary history of those enzymes. But as I said before, what we really care about is not necessarily those single mutations. So where can you introduce diversity to improve the enzyme? But how do those single mutations add up to constructive trajectories? How can we avoid negative epistasis? And basically, in combining mutations with each other, there are three different scenarios, right? We can be additive. Those two mutations are simply adding up, and a twofold improvement and another twofold improvement end up in a fourfold improvement. So this would be highly desirable, and this is something we really want in protein engineering. What is even more desirable is if we end up to be positively epistatic, so synergistic, right? We have a threefold improvement and a threefold improvement, and we are higher than ninefold in our improvement, right? So that's highly desirable. And what we want to avoid is we want to avoid being negatively epistatic. So what we did is for around 8,000 data points where we have both the information on the higher order mutation, right, how to combine mutations with each other, and for the single mutations that make up this higher order mutation, we constructed this epistatic model where we plot the additive fitness, so we would expect, right, to see in a higher order mutation, or what you see here basically in gray, with what we actually see, which you see here on the y-axis. So basically, everything you see here in red is now positively epistatic, and everything you see here in blue is negatively epistatic. And what we then did is we defined this property of combinability, where we pooled everything together that's either positively epistatic or negatively epistatic, and then counted individual mutations in this pool of combinable mutations, and basically, said the more often a variant occurs in this pool, the more combinable it is. The more, the higher its potential for constructing a constructive higher order mutation is. And if you look now at the position in sequence of where you find those combinable interactions, you find that there's this global hotspot at position 241, which is going to follow us also to the AI side of things, and which seems to be a very combinable space of the protein. And if you plot then a network graph of each individual position in the protein, where distance represents distance in the structure, and each edge represents a combinable interaction, you see that this synergistic combinable pool of T241A is actually global all across this protein, right? So we really found here a global hotspot for combinability. So how can we use those profiles? How can we use those maps? We have those 17,000 individual data points. We have a property that we call mutability that we derive from it. We have a property that we call combinability that we derive from it. How can we use that for extrapolation? How can we find even better, mutants simply by extrapolating from that? And the first thing we did was, even before we started collaborating with Simon, simply sitting down and be like, okay, we can do this rationally, right? We can rationalize, okay, and T241 here seems to be very combinable. Let's go all over the sequence and sample mutations that are improving and simply combine it with T241A. And interestingly, in 3 out of 4 cases, that actually gave us a high improvement in catalytic rate, up to 11-fold. And this, interestingly, also was highly synergistic. So in those 3 cases, we were up to 2-fold better than we would expect it to be. And this means that we can sit down as humans and rationalize with certain parameters from this profile of, how should a constructive trajectory look like. But then we asked, can we do this, or can we first automate this with AI? And can AI also tell us something that we as humans, might have a hard time spotting or might have a hard time prioritizing? So Simon constructed this linear regression model, and maybe Simon, you want to quickly share your journey of how you arrived at this linear regression model. But there were a couple of models that you tried before, and this really took us a year to arrive here. Maybe you want to say a sentence on that or something?
Yes. So I could tell you into some of the pitfalls I ran kind of at the beginning of this project, and as I wasn't, I think, enough aware of what was even going on. So I started off just doing a bulk standard training objective against a regression objective, basically. So a squared loss. And then, as we've seen, right, the fitness values actually have a limited dynamic range. And so for the high fitness values, those would actually not be very indicative or not be very confident that they are actually that high. We would just know that it's higher than others. And so actually a ranking type objective is much more meaningful in that particular case. Another thing that I started off with is I looked a lot at like general ranking metrics like the Spearman you saw and so on. And it was very hard to get any signal because it is very noisy data. So it was very hard to get any signal of what does better than anything else with that type of metric in our particular case. And so what then started really helping was to break it down much more closely to what the task we were actually trying to solve was. And so that was breaking it down into two questions actually. One is, is that particular variant likely better than wild type or worse? So kind of breaking it down into a classification task. And so we then used that in our assessment very often as like a hit rate. So we were just trying to predict, is this likely a hit or not? And then another component which would tell us just, is this, how much better is this very likely? And so that started to help us get some of that differential visibility. Another original model we started with actually before we even went through the linear regression. I was looped into this project kind of midway through, and so we were at the time very excited about trying out some big deep learning architectures, language models, and fine-tuning them on this type of data in order to do the fitness predictions. And so what I kind of trusted based on the history of the project was that simple baselines had been built before, but turns out that was not actually the case. So there weren't any simple baselines that had been constructed. And so we were trying around for a while with fairly large, deep learning based models, when really what we should have done is we should have started with a very simple baseline and then seeing what the differential effect is to going to higher parameterized models. Because what ended up happening very often was, because this is a data set that is so noisy, the deep learning based methods tended to just immediately overfit to the noise, and it was very hard to regularize them in our case. And so a model that was much less heavily parameterized, but much more robust, was just much more useful for this particular use case and data set. And so we could have arrived there much quicker if we had actually done the proper baseline. And so that's something I think that I encourage everyone to do, is to start with the simplest possible baseline. What's the baseline if you have 0 parameters, right? What would be your baseline expectation if you don't do anything and just use a smart human heuristic, some basic rule that you can come up with? How well would that do? Then what's a weakly parameterized option as your second baseline, maybe a linear regression or something like that? Always useful to understand what a linear model would think. And then move kind of along the scale. In our case, it turned out that this happened to work the best for us, even. So even the deep learning models couldn't outcompete the baseline, and so in the end we stuck with this model because it was also much more understandable and explainable in our case. So really don't shy away from the machine learning box in the original Venn diagram that I had, purely because in the news you hear a lot about deep learning.
Yeah, and those simple models actually worked incredibly well. So what Simon did is he ranked all individual single mutations in the data sets. And what I told you before is how we generate a library. We use random mutagenesis called error-prone PCR. And we really only have access to 20% of all single mutations in our data set, right? So we know for each and every position around, 4 or 5 maybe different data points. And what would be very useful for us as protein engineers is to go beyond those 4 or 5 data points and learn about the rest, right? And have this as a tool, to augment kind of what we get from an error-prone PCR library. So Simon ranked all individual single mutations with that model. And and gave us the top 5 predictions. We tested those top 5 predictions in the lab, and we found that in 3 out of 5 cases we actually got an improved k-cat, which is the property that we care most about. And what we were really, really excited about is that if we, when we then looked at the top prediction from the model, which is at position 241 again, we found that this top prediction of the model actually outperforms the best mutation in the data set. So what we can then go is, or what we can now say is basically we probe the local fitness landscape around our IRAD. We very, very patchily generate that data, and we can now use that data and extrapolate beyond what we can see in our data set, which is a very, very useful tool for us as protein engineers to have. And in this case, we can go up to a 7-fold improvement in catalytic rates with this T241G variant.
To add to that, Max, there's different directions of extrapolation, right? This type of extrapolation is, given I know something about the chemical space at a given position, what can I say about other chemistries at that position? So other side chain chemistries at that position. So this is extrapolation in the chemical direction at a given position. There's also extrapolation along a different axis, which would be what can I say given I know something about this part of the protein? Can I say something else about an entirely different part of the protein? This particular model is not what was designed to answer this question. Here we're talking about extrapolation within the chemical space. And in fact, this model would not be capable of extrapolating between different sites. For that, we actually need to go to higher order models.
Exactly. And sorry, over there, yeah?
We have a question online.
Yeah, sure.
So Manuel is asking, there's like 2-part question. So the first part is there any noise in the fitness for each sequence? And if yes, does the noise in the prediction match the noise in original?
So noise, yes. There's a lot of noise in the prediction. We can estimate that noise because we have replicates, and we can estimate that using a standard deviation, and that can be up to 3-4-fold, right? Because we are logarithmic here. So that actually is a pretty hefty noise kind of factor that we have in. The second part, does that match the noise that we have in the model? I'm not aware that we actually used the noise as a parameter in the model right now, right?
We didn't predict the noise in this particular case, no. We used it as a weighting. So confident variants go in with a higher weight rather than less confident ones, but it's not currently predicted. Yeah, it's something that we're looking into, though. The way the confidences are obtained is actually a pretty interesting error model based on Ben Lina's work, right? And I think, yeah, that is some really interesting biostatistics that I think was actually really, really useful as well for us in order to understand it. I should have looked at this earlier. Originally, I didn't, consider the noise from those fitness values too deeply. But if you look at the noise of those fitness values, you'll find something like, fitness 2 plus minus 1.5. Fitness 2 plus minus 1.5, what does that mean? 2 means this is a logarithmic scale based on the natural logarithm. So 2 is like factor 9, factor 10. And plus minus 1.5 means, a factor. So this is a factor 10 improvement plus minus a factor 5 or so, something like that. And so really this is very high noise. And so the reason I'm mentioning this is having looked at this, it would have been, I hope it would have been much more obvious to me that ranking mutants doesn't make sense, ranking close by mutants. So really, in order to get signal from this data, the resolution is not high enough to rank really close by mutants. So what you would need to do is you would need to bin it along a resolution that is adequate for the noise level that you have. And then you would need to ask whether you can compare things that are meaningfully different based on that noise. So basically like standard hypothesis testing style things that you would learn in a statistics class, but use that as your metric to judge whether you're making progress.
Perfect. I have a follow-up question for that.
Okay. Yeah.
So when you consider noise as one of the parameters to judge fitness, does it like give bias to your prediction, do you think?
I think it will partly, yeah. I mean, there are a lot of biases in the whole process that relate to this assay in particular. Things that overexpress will always be biased because things that overexpress will have higher counts, both at the input and the output. And so they'll be more confidently assessed because the whole noise model is based on a count-based modeling. And so the higher the noise is relatively to, the higher the count relative to the higher count, your error on that count will be smaller. And because of that, stuff that overexpresses will have a higher confidence and therefore also go in with a higher weighting. Stuff that underexpresses, right, might be fantastic, but it's very hard to tell because we get very few reads based on that. And then I think there's many other biases. And
I think one is, for example, that we simply don't have, data points equally distributed over the whole sequence. So there are just spots where you only have, 3 data points maybe, and then there are spots where you have 7 data points. And the model would be then much more confident at spots where we have 7 data points, but that wouldn't necessarily mean that we would have to look for improving mutations there, right?
And the way we generated the variance is also based on error-prone PCR, right? That will have a bias because you will likely only mutate one of the nucleotides in the codon. And so that limits the chemical space that's accessible to you. So if you have a methionine at a given position, you'll only see a few specific codons at that position. Yeah, that would be another bias. And we're actually, there's a next generation of this work coming up with Matt as well. And he's made a couple of modifications to the workflow that try and, control some of these levels of variability now that we understand them better.
Thank you.
Thank you.
Perfect. All right. So now we talked about, us extrapolating kind of in the chemical space, as you nicely formulated it, right? Now, can we use that combinability data that we generated to actually also extrapolate in the kind of higher order space, in this combination space, right, to avoid negative epistasis? And so what Simon did is he trained this decision tree model and again used a lot of different features for that model, for example, from ESM, pseudolog likelihood, some basic elasticity information. Maybe you can elaborate on that later in the kind of interest of time. He again came back to me and was, okay, I ranked all the double mutations, and those are the top 5 predictions of our double mutations. Let's go test them in the lab, right? And this model performed even better than our single mutation model, and we were actually able to see 4 out of 5 improving variants when it comes to the most important property k-cat here. And what we were very pleased to see is that actually the model picked up on the fact that maximizing combinability is very, very is important and is an important feature of the data. And it chose to combine T241A with other mutations in the data sets to find those highly improving mutations here. And in 2 out of 3 cases, those highly improving mutations actually, again, were synergistic, positively epistatic. So what this model now allowed us really is to go beyond the single mutation space and go to the higher order mutation space. Do you want to add something here, Simon, or do you feel like we can move on?
Maybe one thing we did was we were very skeptical of how well this would work because the double mutant space is just so much larger. And also because of the just pure level of noise. And so I think the whole double mutant space for this particular enzyme is on the scale of 1-something million possible options. And so we only got to test 5 because running all of these assays in plates is actually a lot of work for Max. And so we were thinking what allows us to also maximize the chances of success. And so we artificially restricted the design space in this particular case in order to make sure that we have a better sampling. And so what I mean by that was because we were skeptical at the initial capability of how we would generalize on the double mutants, we just picked the top 5 double mutants among the set of double mutants for which we observed each single mutant in the data set. And so that rather than, I think, the 1.5 million, that's like still 500,000 membered space, something like that. And so that's from within where you see those mutants picked.
Yeah.
Just as a qualifier.
Maybe a super naive question. Would you also try to predict triple mutants?
We didn't then. I think, I mean, it would be something that we would be very excited to do. I think we would need to do that in the next revision of the approach, because then I think we'll have more confidence in the fitness values of the individual mutants. And I think that will help us massively. Because if I do think if we have more confidence in the fitness values and less noise, we'll start to be able to tap into more of the heavier parameterized models and use them. Here it was used as a feature to the model, but we can actually use the parameters of these more heavily parameterized models directly if we're not at risk of overfitting as much. And in order to avoid that, we need to bring down the noise level. I think we're thinking about doing it for the next iteration, but we didn't do it for this one.
If I may have a follow-up.
Yeah, absolutely.
When you do the triple mutant modeling, would you also take use of the single mutant and double mutant data to train it, or would you just purely use the triple mutant?
We would probably, yeah. We would definitely try and use the single and double mutant data as well, yeah.
All right. And then maybe as the last point here of our little case study, we then, just combined all those different tools together, right? So we basically saw those models that Simon generated as protein engineering tool, to expand the chemical space first, to then, go to double mutations and find constructive trajectories. And we combined it with our, rational engineering approach, where we simply went and combined hotspots with each other and just pooled those approaches. And that actually gave us the best outcome. What we were able to engineer with this pooled approach is a 16-fold improved KCAT to KM variant with a 23-fold improved KCAT. And I think what this basically shows us is that we can, go for a 4-fold improvement in what we see in the data, right? And then we can then go 1 order of magnitude higher, with our best engineered variant. If we pool all our efforts, if we pool the rational engineering together with the AI, and actually, have this broader approach. So basically, us as human protein engineers in this case, we're still very, very useful to go and to use those models, right, in a rational way. And maybe a hand over to you quickly here to kind of demonstrate that what we did here is explainable AI, and that we can then rationalize actually what our model actually did.
Okay, yeah. So what we wanted to understand was how much of this could have been done by just using, say a language model like ESM, without using any of that actual assay labeled data, and how much of the importance of the predictions of the model actually came from having that assay labeled data around. And so because the models we used have associated explainability techniques, so linear regression, obviously, a lot of techniques available for that, and then decision tree based models, in this case it was a gradient boosting type model, also have very nice explainability work on them. We could use that to post hoc, so retrospectively see what the effect of the different elements of the model were on the performance. And on the right hand side in particular, you can see feature importances that were used. This is the case for the double mutant model, and you can see fitness, min, combinability, and additive fitness. Those are all 3 features that are derived from the data set, from the assay labeled data in red, and you can see that those by far had the largest impact on the modeling results. And then I guess in a 5th position, we actually then get the language modeling features, which gave some general, I guess, consensus. I mean, what we found actually, we also did, you did take 5 mutants from ESM to the lab as well, and they actually also gave us improvements. I think, what was the largest? Threefold?
Threefold or something, yeah.
Something. So the best mutant that was predicted by ESM was threefold improved. And funnily enough, when you looked at the predictions, those would actually all be back to consensus mutations based on an MSA profile. And so these language models are essentially, I think a mental model for these language models is just that they give you the information you would, or similar information to what you would likely get if you did MSA-based modeling. And so we included them as well in order to have information from evolutionarily related sequences. And so you can see ESM fitness also played a role, but in the local fitness landscape, it was more important to actually get the fitness data from the assay context than it was to get the information from evolutionary relations. And on the left-hand side, you see a learning curve. And so that was just for us to understand the impact of the differential increase in data on the performance of the models. And you can see on the bottom axis the training set size grouped into different well-played sizes in order to have a rough comparison of if you had this many data points in a 96 well-played, et cetera, what would the likely performance be? And so this was just a simulated experiment where we'd always draw multiple replica. I think I did this in 10,000 replica of data set, of samples from the data set, and understand kind of how is the impact of scaling data on training performance. And you can see the typical logistical curve because the x-axis is logarithmic here. You get logistic kind of improvement of downstream task performance with training data set size.
Nice, yeah. So this brings me to the end of that case study, actually, and just to highlight again, if there are any enzyme engineers in the room, don't throw away your data. Your data is really useful. Go get yourself a collaborator like Simon who can make that data use and basically accelerate the time you need for your protein engineering campaign. In this case, that was a 4-fold improvement that we had in our data set, which was already pretty large for enzyme engineering standards, and we could then add another 4-fold to that by adding Simon's AI models to the picture. And maybe with that, I'm really going to hand over to Matt here. Just to give you another quick, teaser of, AI in the enzyme world, but from a completely different perspective, from the enzyme design perspective. And let's quickly go. Maybe any questions or thoughts on kind of that part right now? Yes, over there.
I had a question regarding the feature importance work that you did. What was the application of this? Did you actually go back and refine your model and take away some of the features to improve the computational power of it in the end?
We did do feature ablations as well. They weren't shown on the plot. I think one, actually, the left-hand side plot showed an ablation of ESM as well and showed that removing it didn't harm performance or only in a tiny bit. We used it mostly to get confidence that the assay labeled data was actually useful and that we couldn't have done that if we had just, modeled without going into the lab. So that was our main use of this. I think in the future, I would probably do slightly different analyses in order to do a more prospective kind of what would we do as an design type of thing. This was more retrospective, kind of understanding the contributions to the model, like SHAP values, so on. I don't think I was super prospective. What I think was a good source for us in terms of understanding how to improve was actually doing a step-by-step walkthrough through the assay and trying to write out what are all the sources of bias and noise that enter at every stage. And the two main bottlenecks are the sorting step, in our case, right, the microfluidic sorting step. I think one thing we will definitely do for the future is oversampling at a higher rate, because we found that when we did the error modeling on, what is the error we observe between the replicates of the experiment, we find that the primary source of error is multiplicative, which is indicative of, so multiplicative means it scales with the number of counts. And that's indicative of a problem on either the bottlenecking side or the sequencing side. And so we think the most likely option, because Max then did a fantastic analysis also on PCR bias and PCR noise, which would amplify any thing that we get out of the sorting. And we think the oversampling at sorting time is the single largest contributor based on that analysis. And so that's one of the improvements we're making. Another is controlling for expression. That was a bias you've mentioned earlier. So those are some of the thoughts, but they didn't really come from this particular interpretability analysis. This came more from going over the workflow together and looking at the modeling of the errors in particular, and trying to understand where they likely come from based on the profiles we're observing.
Thank you.
All right, perfect. Then maybe over to Matt. Next slide.
Okay, thanks, Max. So I'm Matt. I'm also from Max's lab, and I am a wet lab person. And I just want to talk very quickly for just 5 minutes about a slightly different perspective, and mostly focused around the kind of iterative cycle of working between the wet lab and the dry lab. Because I don't know if it was highlighted before, but we don't actually work in the same place. So our dry lab is in center of Cambridge, and the wet lab is in West Cambridge. Sorry, the dry lab's in West Cambridge, and the wet lab's in central Cambridge. And along with that comes a lot of kind of unique communication challenges that we need to overcome. And then you compound that with the difference in background and mindset of a typical wet lab, dry lab scientist. We find that there's quite a few, hopefully, things that we can discuss today a little bit and learn from each other, and kind of, before they become a problem, turn them into something that we can really draw the two perspectives together to build something quite unique. So all of this is in the background of a project I've been working on, along with Simon and Kieran and Max and the Baker Lab in Seattle, and it's around enzyme design, in particular de novo enzyme design, using RF diffusion as the kind of workhorse. So essentially the background is my wet lab experience comes from enzymes for processing biomass, waste biomass, and using enzymes to extract useful compounds from that waste biomass. The class of enzymes that I work on are glycosidases, and they essentially depolymerize biomass, which, if it's plant, is a very high proportion of polysaccharides, so long-chain polymers. And the enzymes I work with turn them into short-chain or monomeric compounds, which you can then use as chemical feedstocks, or literally as feed, because, of course, sugar is food. So the AI part of this is the de novo enzyme design, of course. So the challenge that we are faced with is we have a existing large number of glycosidases that are known to exist from the environment. These come from the metagenome and also have gone back kind of tens of years in terms of experimental characterization of these enzymes. And we want to generate totally new ones with one day the goal of making them tunable in a way that the existing sequence space at the moment, we've already explored how we can tune that. Let's tune a larger sequence space. So based on this de novo design, then processing to microfluidic screening of this library of de novo designs, followed by this NGS and hit identification or active learning that Max has already touched on. And so from what matters to the wet lab scientist and what matters to the dry lab scientist in this particular project are, in the wet lab, we have to consider the target molecule that we want to be working on. So is that the actual biomass itself? Is it something purified from that biomass? Or is it an analog, a chemical analog of the biomass? And that is incredibly important because that will feed into what the dry lab uses for their enzyme design. And then also in the wet lab, we have to consider the screening method that we're going to use. So of course, microfluidics is the method that our lab uses. And actually developing an assay in microfluidic format is not always trivial. So that is also going to be another consideration when we're designing a project together. And then, of course, what is the biological question we're trying to answer? Or is it more of an engineering problem? Are we trying to understand how glycosylase enzymes work themselves? Or are we trying to build the biggest and best, so to speak? And then from the wet lab point of view, of course, this isn't my point of view, but from what I hear, what kind of tools do we have available for design? Are we going to be making our own tools, or are we going to be slightly repurposing tools that already are known to work well? And then what kind of compute we have, of course, and then what is the likely hit rate from this whole pipeline going to be? How much work am I, as a wet lab scientist, going to have to do in the lab to scroll through designs until we find one that really works? And what slightly sets the project to slightly different to the one Max just described is that it has started from a place of both the wet lab and the dry lab have been communicating from the beginning. So we're aiming to design this project in a way that kind of matches all of these two things together, which so far we're finding has been very important. So how did this collaboration come to be, and what have the challenges been so far in communicating between the two sides of the lab? So Simon actually came and presented to us in one of our wet lab meetings and presented the tools that exist already for enzyme design, which we were kind of mildly aware of, but Simon helped to put it in a kind of understandable perspective for us, and expressed a desire to apply these tools in some way and to work with us, and actually said this explicitly, which meant we had a clear aim to collaborate. And his explanation was very at our level, perhaps luckily, because going into a room full of people, you don't know what level you're going to need to explain things at. But maybe in this case, this was the kind of luck that got the project started, or maybe it was skill on Simon's side, or both. And then, of course, afterwards, he was keen to discuss proposals for how we can combine our two sets of experience. So, of course, at the very beginning, we have how do we make that initial contact? How did we make that initial contact in our case? Because I find sometimes talking to people, this is a bottleneck in collaborating between the wet lab and the dry lab. How do you find a wet lab that's interested in testing your designs, or how do you find a dry lab that's interested in analyzing your data? Unfortunately, I don't have a great answer for you, but in our case, we were already collaborating when I joined. For years, there's been an ongoing collaboration between our two labs, but maybe this is something that we can discuss later in the workshop as to how to maximize these opportunities. A session like this, for example. And then with explanation, how do we convey the state of the art without trivializing it to pique the interest of the, in our case, the wet lab scientist, while without overdoing it with the models and the exact specifications of what's being done on the dry side. So the action out of that was that we used the information from that meeting to select some suitable targets, or targets we thought were suitable at the time. So this is going back to which part of the biomass molecule are we going to be doing the design for? Is it the actual thing? At the time, we decided to do a purified form of the actual bit of biomass, and then presented the proposal that we had for the enzyme family we wanted to work with and the substrate back to the AI team in the wet lab in another small meeting. And at that point, we also had another potential communication problem, because the question of whether we yet knew enough after one meeting to present a plausible scenario back to the dry lab again for them to design was definitely up in the air. And as it progressed, it turned out that the first design, the first suggestion for the project was not, in fact, the best one. And as we moved on, we actually moved past the first target that we picked. Otherwise, we could have been stuck in a massive backlog, trying something that wasn't going to work. And then, of course, after trying out a few proof of concept designs, the AI team fed back to us what their designs look like and kind of leveraged the eye of the wet lab. So asked us to physically look over the designs that they'd tested with the, or predicted based on the very small proof of concept experiments, and asked us to assess how reasonable we thought these predictions were. And this is a it's a test of confidence in both sides, because on the one hand, as a wet lab scientist, I have no idea how good my eye is going to be in assessing something like this. And I imagine on the dry lab side, you have to trust that the wet lab scientist has some extra experience you don't have in this kind of magic eye for assessing design qualities. And another question would be if the wet lab scientist is giving feedback that to the best of their knowledge is good, but actually the model has seen something better, the way they're missing out by limiting to what the wet lab scientist says is the best or reasonable or not. And then, of course, the final one, standard in any project, is at this point we were able to put some kind of project timeline together, estimates in place based on all of this cycle that we've gone through. And then we had to, of course, accurately convey timeline information between the two sides of the group. So yeah, that was our kind of some of the challenges that we faced. And we find that some of the best ways to overcome these challenges are these small informal meetings rather than the big lab meeting style presentations that require a lot of prior preparation, can take weeks to prepare for. Extremely informative and important, but for keeping momentum between the wet lab and the dry lab, we find the small meetings, in our case, have been really effective. And then consistent and honest communication between the active members of the project about hurdles and progress. Another big source of this, a big point about this is about noise. Really, as Max said before, and Simon as well, accurately communicating how much noise the wet lab scientist feels is in their assay. And this can help point out problems that maybe this set of the data is too noisy, but we should really focus on the set that we think is good, rather than saying, give you the data and, play around with it, and then you find out down the line it's a bit noisy. It saves everyone a lot of time. And of course, incredibly important, I put relatively infrequent, but still frequent, just relative to the small meetings. Meetings with the PIs, of course, to align exactly what direction we are keeping the project in, to make sure that it's still within the kind of the grand scheme of what's important and what's worth doing. So yeah, with that, any questions or thoughts? And if not, I think tea.
Awesome, yeah. So maybe before the tea, we thought for the last, I think half hour we still have left. We might combine the last, coffee and tea break and just, make a bit of a more interactive session. Which is really nice is that we have already groups kind of seated around the tables. So maybe everybody can get themselves a coffee or a tea and then come back to the space. And then we've prepared some questions. And maybe you can chat about your own research project. Do you have the questions out? Maybe we can quickly read them now, or
let's do it when people got there.
Perfect.
Coffee.
And then grab some coffee, and then we reconvene back here. We talk about our own projects and have a bit of a more interactive last half hour. Perfect.
to answer. Then what data is available to you, or are you planning to collect if you haven't yet collected it? And then in particular, I think that's the one to spend most time on. In your mind, what are the most important considerations when working with your problem and data that you have? And we thought what tends to work best is groups of like 4 to 5 people. So I guess that gives us 2 or 3 projects we can discuss. If there's people who are kind of keen on getting others' inputs on their project, feel free to raise a hand, and then we'll just form, I guess, 2 or 3 groups, 3 groups probably, around each of you, and then we'll just join as well for the discussions.
And then we maybe use the last 5 minutes to kind of announce what conclusions you made, and maybe give a quick summary of what the project is, and what the considerations are that you thought about. Only if you want to, obviously, but I guess 1 or 2 of you will just quickly share with everyone else what they thought about. Yeah.
That's good. Any volunteers who would be interested in getting the general feedback of the group to your project, your data? I guess we'll just do first come, first serve. So feel free to just raise a hand. 1. We've got space for 2 more. Anyone else willing to volunteer? 2? 3. Okay, wonderful. Do you just briefly want to say 1 sentence about it so people can go and find your table? Oh yeah, wonderful, thank you.
Again, I haven't started anything yet, but I'm going to start next Monday. So, so basically the project will be working on codon expansion. So basically adding a natural amino acid for protein design. So what I'm interested to see is I don't really know what kind of data set. I'm coming from a wet lab-based scientist, so I understand the biological side of things, but I want to explore more in between.
Okay, awesome, thank you.
I'm more interested in how, by looking at the human gut microbiome data, we can try to find patterns or associations with the human lifestyle, or like, for example, medication and other stuff. I am a bioinformatician by training, and I did some wet lab as well. So my main concern is the data is so noisy, and how am I gonna deal with it? Thank you.
Okay, nice, thank you. So we've got microbiome, protein engineering.
The problematic, or my problematic, is very similar, and basically, since I work on many genomic sequences, I would like to like somehow make this data set more equilibrated, so I can have examples of many instances of the phenomenon I have or that I am studying, but sometimes it's really hard because normally genomic data sets are pretty unbalanced, and so I would like to know if somebody knows how to tackle this problem.
Okay, awesome. Nice, so we got genomics, proteins, and microbiome for 10 minutes. Feel free to just join a table, anyone else, and we can have a very brief discussion. I'll quickly type in the questions on the slide, and then we can maybe show the slide in the room. And then we'll just join one of the projects.
1 minute to wrap up. So 1 minute of discussion, and then we just wrap up quickly. Yeah. We wrap up now the discussion. Maybe we wrap up the discussion now. All right. So perfect. So if everybody that was involved in the discussion, maybe if we could have, should we do it from 1 or 2 tables, or maybe all tables? Like a very quick summary of what was your question, what was kind of the the data that you kind of thought about involving in your question, and kind of other considerations that you discussed, and then we give a microphone around maybe quickly to also make that available to the online audience. All right, where should we start? They have the biggest group, all right.
Well, to be honest, we just got to the first question, to the what question are you trying to answer, because it's a little bit hard for me to explain or make understandable this is, because I just understand it, and yeah, we just got to that point, to be honest. But actually, they gave me some ideas, and it was really helpful.
Great.
Thank you.
We were talking for very long.
Time constraint is our fault.
Thank you. Yeah, we discussed all 3 questions, and we agreed that there are many issues to deal with. And yeah, probably sorry, I don't get your. Katharina said maybe I can look at some literature to see if something is already published, which I will keep doing. And Max, yes, he suggested to see if it's possible to generate more data in the lab, which I think it's tricky in short term. And also for the available data set side, it's a bottleneck for me personally, because whenever I want to access some data, because it's human gut microbiome, I need to fill a lot of paperwork, and it takes forever to get signature. I don't know how to solve this, but yeah. Thank you.
Perfect, thank you so much. And then the last table here.
I don't think we have much conclusion, to be fair. I think it's a brainstorming idea that where we can start protein engineering using codon expansion, and chemical properties is complicated. I just heard from Simon that there is a database that has 360 properties of chemical properties you can play with. So interesting insight, and a lot more to work with. Yeah.
Perfect. All right. Then I think we wrap up here. Thank you all for coming, and yeah, have a nice evening.